welcome back to the lecture series in cell culture technology. So, today we are into the 5th lecture of the 2nd week. So, we are into now week 2, lecture 5, W2L5. Okay. So, in the last class, we talked about that in this corner, if we put a laminar flow hood with a sitting arrangement like this, okay, or sitting arrangement like this, or maybe a sitting arrangement like this, we cut the. So, before laminar flow hood design, let us decide one more stuff. You get the animal inside, right. So, here you are getting the animal inside the facility. Now, you have to have a setup for sacrificing the animal. So, you are sacrificing the animal means you have to kill the animal, and in that case, say for example, we consider that you will be using mice or you will be using rats or something. So, you remember in the last class I told you that you needed the animal ethics committee's clearance. So, that is where you have to get your clearance. How? First of all, that clearance will come with how many animals you will be using, 1, where you will be housing the animal, 2, in the housing what all things will be providing in terms of food, the kind of food you will be supplying, the water supply, the air conditioning and all those paraphernalia. But what is very critical is that how you are going to sacrifice the animal. If you are sacrificing and taking out the tissue, what kind of mode are you going to use? Are you going to kill them by carbon dioxide, or you will be doing something called cervical dislocation, okay, or you will be using some other mode. So, there are different modes by which you can sacrifice the animal, and these different techniques are getting revised and updated from time to time. Through the years, there are several improvisations which have taken place, and the veterinarians or people concerned are aware of it, and they can literate you on that this is the technique which is best suited for you, or for this kind of animal or that kind of animal, or this kind of, or say for example, you wanted to do an anesthesia. Say for example, you are not sacrificing the animal, let us put an alternative. Okay. You want to do is an anesthesia, because say for example, you wanted to implant something in an animal, and you want to take the surrounding tissue. So, in that case, what kind of anesthesia you are going to follow? See, if you are not a veterinarian, you will not be aware of it, because there is no reason for you to know this additional piece of information. So, this part is very critical, and whatever you do should tally with what you have promised with the animal ethics people. There should not be any discrepancy. You promise something and you do the stuff a different way. So, let us assume in this situation, then you wanted to do a CO2 sacrificing. So, that again brings us that you needed a supply of CO2, which most of the labs currently across the world does, So, and you have to have a chamber, that is kind of a chamber like where you keep the animal, and you have to have a gas supply something like this, and you allow the animal to have a peaceful death, and then whatever way you wanted to dissect it out, are you decapitating the head, or you wanted to remove some kind of a gut tissue or some kind of muscle, or you want to do isolate the cardiac tissue, you want to isolate the brain. So, the rest will follow, but this part is very important. So, in this facility you should have a small corner and prefer a corner and close to the sink. The reason being because this is something which is kind of you wanted to do it very cleanly. You don't want to mess up things here. Okay. So, you have to have a facility. In the case, of course, these are all assuming that you are doing primary culture, because if this is clear to you, then the secondary cultures and all will be very easy to you. Okay. So, you have to have a facility somewhere, and this animal sacrificing should be deep inside the lab, okay, even in the outer facility. You should not have something of that kind out here or somewhere out here, right. You prefer to have this very close to the door, okay, somewhere out here. Or some people, what they do, which I do not know, I mean how good it is, they even have something outside. They just sacrifice the animal and just bring the sacrificed animal inside, instead of doing the sacrifice inside the facility. Well, I mean it has its advantage and disadvantage, because the disadvantage is that then you need another additional kind of enclosure, which is much more exposed, and the disadvantage is that you do not have to do this stuff inside the outer facility of your lab. Okay. So, I will leave it to your wisdom which one you prefer, but some such facility has to be there, and mind it, this will never be written in textbook how you do it, and this is what I am telling you is from my experience of setting up labs, design and layout, how you really have to think beyond. Because when I set the first lab, I still remember back in probably 1997 or maybe earlier than that, I was not aware of this, and there was no textbook which could tell me. So, it was more of like and this was not the www.ida of internet and all those things. These were like far cry. And so, there are a handful of textbook here and there, but not really telling you how really you set up the whole thing, what are the points you have to keep in mind. So, you can see it was very trial and error, talking to people and seeing the facility and figuring this is not the right way, this is like So, whatever I am telling you is from my experience. There is no textbook which is going to tell you all these things, that how you should, but you have to think in advance, because if you think in advance, I am pretty sure you can think far better than what I am putting out here for you. Okay. And if you think it right, and there are people who are very good, they can tell you that if you put it here, this will be a problem. They may not be knowing your science, and they do not need to even, but they can give you a better insight how you really can design the whole thing in a way. What I am trying to tell you are those rate limiting points which most of the time we missed out, and then we do some hanky panky job, oh, do it like that, fit things like that, and that is really a bad design, bad way of like putting things together. So, please be careful, whenever you set up the thing, think calm, quiet, think over it, how you really want the facility, because if the facility looks nice, then you can bring people, and finally, the end user will be happy, this is a very systematic facility. And one more thing, these kind of facilities takes a lot of soul to be put there. So, those whosoever uses such facilities should carry that spirit of that soul, that we are not going to mess up with, because any negligence could destroy the work of many other people, because you are handling biological samples which are prone to contamination. They can destroy your whole facility in no time, and once the contamination picks up, it is very tough to contain them. Okay. So, coming back, so you have to have an animal sacrificing facility, and you take the tissue out, whatever concerned tissue, and now you have to do everything in the hood. Now, when you get into the hood with the tissue, you have to have a buffer system. This part we will come in the medium development. You have to have a buffer system which will be helpful to ensure that the tissue remains alive during your dissection part. Okay. The animal is dead anyway, but it has to be supplied with necessary. Sometimes if the tissue is very prone to oxidative damage, you have to ensure that how you can handle it, what kind of additional material you may love to put, some kind of antioxidant and whatever, we will come later into this part. Well, we will talk about medium developments, but keep this in mind, this is very important. So, out here inside this laminar flow hood, you may prefer to have a permanent fixture or not a fixture, a permanent location for a dissecting microscope. You will be needing this pretty frequently. If you are dissecting big animals, you may not need it, but if you are dissecting embryos or fetus, you will be needing it. So, see through the catalogs once we come about the microscope and get a good quality dissecting microscope. And here is my word of caution, do not, because this dissection process, especially for the embryo, is a tricky process, and do not spoil your eyes by having a bad microscope for that purpose, because it is a pain, because you will not be able to locate the tissue right if you have a bad microscope. Look through the good microscopes of Zeiss or Olympus or Leica. So, these are some of the giants in the field, or Nikon as a matter of fact, to have a good dissecting microscope. And if you are really good in that, then I will recommend you something which is a very good teaching aid, is that many of these dissecting microscope comes with an attachment to put a camera which could be put on a screen and you can record it. So, let us think of a situation. Your lab works on, say, hippocampal neuron. Hippocampus is a very specialized part of the brain which is involved in learning and memory. And most of the people who work on hippocampal culture cannot rely on cell lines. They isolate hippocampus mostly from rat or mice embryo. This is a standard practice. Once we will come for the primary cell cultures in depth, we will talk about it. So, now in the brain, isolating hippocampus is not something very easy. So, the structure is something like this. If you look at the structure of the brain, so for example, you take out the brain, you remove the skull and everything, you are having the dorsal view or the top view of the brain, you are seeing the. So, what does that mean? That means that animal is sitting like this, and you are seeing the animal from the top. Okay. So, now that is called a dorsal view. So, this is the dorsal view of the brain, and the ventral view, if you kind of twist or make the brain upside down like this, lying this, if this is the dorsal view, right, and you just look it like this, you just make it upside down. So, if this part of my hands are dorsal view, then these are the ventral views. So, if you roll it down like that, the ventral view is something like this. Of the brain. Okay. Now, hippocampus is an organ you won't be able to see from the top unless you cut it like this and you remove this part. So, you have to scoop it out, then underneath this you will see a, so there will be an organ which will be sitting like this, and it is deep inside. This is not something, unless you are very thorough with the brain anatomy and you really can, it is not so easy to isolate this. This is where the hippocampus is lying. So, coming back, why I mentioned this. So, say for example, your lab has been working on this area. So, somebody is an expert who can isolate hippocampus, but this gentleman or this lady is going to leave the lab. So, what do you want when they dissect? Why don't you record it? So, you have a system, or this could be used as a teaching aid for practical training of dissection and isolation of a specific kind of tissue what you will be using in your culture, right. So, it is a good practice to have a good dissecting microscope with possible attachment for camera and a viewer, and this viewer could be even computer. You can interface the camera with the computer, and on the computer screen you can see and you can record it. If there is a good dissection, you can record it, and that could be a tool for cell culture facility, that this is how we isolate the tissue. Okay. So, I will recommend careful while buying a dissecting microscope and go for, like I personally have used Zeiss, they are really good ones, really good ones. I have used Nikon, I have even used Leica. So, these are some of the good ones. Olympus I have not used, but I have heard good things about it. So, but try to get a good dissecting microscope, my first suggestion. So, you have to have a dissecting microscope inside out here, where all your dissection will take place. In the corner, you prefer to have a tabletop sterilizer. What is a tabletop sterilizer? So, for dissection you will be needing dissecting instruments. Okay. So, be careful about the dissecting instruments. Again, a tool which you will be using very frequently if you are a primary cell culture lab. So, try to get the best quality of it. There are global companies which are really good, like Fine Science Tools. Fine Science Tools. This is a Swiss company. They are the maker of some of the finest tools I have ever used till this date. And if you do not find them, and if you cannot afford those ones, if your lab is not ready to afford those ones, you can always go to the local goldsmith, and they can really carve out, or the watchmakers, they can really carve out very nice instruments for you. But you have to pally up with them, you have to tell them that this is your requirement, because most of the watchmaker in our country, they use very fine fine forceps, scissors. So, you can get those things fabricated, which is not a big deal, trust me, or you can get a coarse one and then you can gold plate it and make it sharper, and several things you can do. There are a lot of zone for innovation, tremendous zone for innovation what you can work through, but you have to think how you really can make these things happen. Okay. So, get the best quality dissecting instruments, and there are dissecting instrument box is something like those of you have seen the lunch boxes which are made up of aluminum or steel or something. These are box similar to that. Underneath it you have a kind of cushion like thing which is polymer or rubber kind of thing. You keep a rubber on the side, so that say for example, you have the box like this with a cover. So, you have the rubber bed like this. This rubber bed ensures that the instrument which are there, so you have see this fine sharp forceps, you have very fine should say very fine scissors, scalpels, and you do not want these to get damaged. So, what do you do? You can even fabricate, you can get a box and you can you can put very nice rubber lining. So, even if the tip of the instrument kinds of get hit, should not get damaged. So, again these are the experience which is telling you, because I have seen things getting damaged, destroyed just because we did not take proper care, which you could have done, right, if you have been just a little bit of farsightedness. So, you have to have a dissecting box. So, before you get the dissecting box, what do? You sterilize it. So, you have to have one more thing, you have to have an autoclave somewhere out here. Now, this autoclave can be outside the lab also. So, autoclave is something like a pressure cooker, where at high moisture and high pressure you sterilize anything. So, you will be needing autoclaves, and ensure that these autoclaves are according to the requirements or number of end users. You do not want to get a very huge ones, which is kind of tough. There are modern days facilities where they have centralized autoclaving. So, just before I again get back to the dissecting thing, let me talk about this autoclave stuff. So, it's the autoclaves could be multiple types, like you could have autoclaves like this. You will see in any of these labs, they will have like this huge autoclaves sitting on the corridors. You will see most of the time on a stand like this. Okay. So, they are bolted on 3 or 4 side like this, and they are connected to the power supply. Okay. And here you keep your stuff, and there is a pressure gauge which ensure the pressure, and from here the water kind of creates the vapor. So, in a high pressure and in a vaporized environment, you sterilize the stuff. So, whenever you are keeping anything for sterilization or autoclaving, you put them in a autoclave pouches. There are pouches which are available. So, it is kind of you must have all have seen envelopes where you keep send our letters or something. It is almost similar to envelopes, and you can seal them. So, they come like this, something like the envelopes like this, and this is a sealing. When you seal them, so there is a spot you will see after a certain point of heat and moisture, the color of that thing changes, which indicates that this stuff has been autoclaved. And if you visit websites of autoclave bags, you can see these kind of autoclave bags. They are really very interesting stuff. Explore it online, go online and check it out. You'll really enjoy seeing them. There are a whole different range of autoclave bags which are available. Okay. So, you'll have these autoclave bags. So, whatever you want to sterilize, you keep them inside. They come in different size and shape, mostly different size and shapes. Okay. So, you can have bunch of them already purchased for your facility, and you keep your stuff in them and you bring it out till you reach on the laminar flow head or just close to the laminar flow. You do not open it. Okay. So, you get these instruments autoclaved and bring it inside your facility, right. So, you are out here. So, now as I told you that either you can keep the autoclave outside, or you can keep a tabletop autoclave somewhere out here, especially for this kind of facility, which I personally prefer a tabletop autoclave, which is in your control. It is not outside, it is not bothering anybody. You have a small tabletop autoclave, and these are all available. They are of kind of like 1 feet size or something like this, and they are pretty cool stuff to work with. It does not create much problem. So, close to the sink, because you have to fill water and everything, and they need a power supply, and that is all you needed. So, you get this instrument inside, and while you are dissecting, automatically you may feel like if I could re-sterilize, because from one animal to second animal, from one embryo to next embryo. So, for that you need dry sterilization, which is the tabletop sterilizer. So, these are sterilizers something like this. They are small, like quarter feet stuff like this, and there are lot of glass bead kind of stuff in them, and you keep the instruments in it, and you have a panel like this, and it is connected to the power supply. Okay. And this dry sterilization takes around 90 seconds to a minute. So, while you are working, you can re-sterilize, but there is a difference. This is where you have a wet sterilization, which is far more long term stuff, and here inside the hood you have dry sterilization, where you are only utilizing the heat, you are not utilizing the moisture or the pressure into the game, right. So, you have the dry sterilizer sitting out there. So, you keep your dry sterilizer there. So, while you are dissecting, you can always re-sterilize it in 90 seconds, and still you can use. So, these are some of, and of course, you wanted to have a bunch of pipettes, which most of these labs used as dedicated ones for this kind of dissecting hoods. Generally, I personally used to prefer on those glass walls on both sides to have the pipettes can be kept somewhere like this. You can have system where you are not exposing them out, and you have sterilized pipette tips which are kept inside, something like this. You have the pipette tips sitting there on the bench, which time to time are sterilized, and every time you use, somebody comes with a fresh pipette tip box, and kept it there, and remove it and again replace it with some new pipette tip box. So, these are some of the basic things what you prefer to have inside the dissecting microscope or dissecting laminar hood. So, let us summarize what all you need to have. You need to have a microscope first, a dissecting microscope, of course, if possible connected to the camera, and a viewer, the computer or some CCD device where you can monitor. Second, you have to prefer to have a dry sterilizer. Third, a dedicated pipette and pipette tips sitting out there. So, now we added one more component into it, the laminar flow hood in their outer facility. Okay. So, and where you wanted to put the autoclave. So, my preference will be have an autoclave inside a small tabletop. You can have a big autoclave outside, maybe on the side of the corridor or somewhere, but a small ones out here may not be a bad idea. Okay. And you have the water supply, we talked about it. So, from here in our next class we will further build up the story how this whole facility will eventually look, because we have just started on the outer layer, and there are a few other things what I am going to highlight here before I move into the inner chamber or the inner culture room, where other things will be taken care. Okay. So, thanks for your patient listening. Once again, we will continue in the next week further development on these layout, design and instrumentation. Thanks.